Good afternoon, everyone. I'm here today with my good friend Max Peterson. He is a cancer researcher at Brown University, and we're super happy to have him here today. Tell us a little bit about what he researches, and what the day-to-day life of a cancer researcher is. Thanks for coming on, Max.
All right, thank you, Michael. It's a pleasure to be here talking to you and tell you about what I do.
Awesome.
So, as you said, I'm a cancer researcher at Brown. I'm a PhD student. So I'm in my third year. So I'll hopefully have a PhD in like maybe 1 to 2 years, something like that. Depends on how the research goes. So I study a certain type of blood cancer called myeloproliferative neoplasm. And this type of cancer is only really seen in older people. So I'm studying cancer and aging and how the two are correlated. And so we look at the level of protein signal transduction. So a protein is kind of like it's the basic functioning unit of a cell. So if you have a drug, like an anti-cancer drug, it's targeted to the protein. So if you can shut the protein down, then you can shut down the process that causes the cancer. And so I'm studying how proteins signal onto each other and hopefully identifying new ways that we can target those proteins to treat this type of blood cancer. And we're looking at aging in particular because the protein signal transduction, it changes with age. And we want to look for those changes in particular because maybe that would tell us a bit about how how everything is happening and how the cancer arises. Um, so I guess like a good analogy. I think analogies are good ways to describe everything. So a good analogy would be like I'm kind of like a mechanic, but the car was not made by people. It was made by just evolution and time. And so we don't have an understanding of this car because we didn't make it. We have to go in and we have to figure out which parts connect to other parts and figure out what they do based on if we take something away, for example, how does that change something else? And so, but the trick with being a mechanic in molecular biology is that you can't actually see anything and you can't hear anything. So you can't hear if the engine is running funny, and you can't see that you have a flat tire because everything is just so small. So we have to use these indirect ways. And so, like a large part of this PhD training is because the PhD is you want to do research, so you want to come out of it with a body of research, but it's also training at the same time. So I'm still in school, really, and so with this PhD training, I'm learning uh, not only the different tools that I can use to figure out, what the different components of this car are doing in the cell, and but also I'm using those tools to actually answer a research question, and that research question is uh, essentially why do older people get cancer more frequently? Sorry, that was really a
No,
long-winded
that
way of
was
saying that.
that was awesome. Thank you. That was fascinating.
Okay.
I'm interested now. And then
Oh,
so
cool.
yeah, and so what is your day to day kind of? Do you like do mostly research? Or are you like in the lab messing around with cells? Or
Yeah. So like right now it's just at home because everybody's quarantined. So
fair enough.
only essential research is going on. So like we have mouse colonies, for example, and
Mhm.
so we have the people who take care of the mouse colonies. They're still going in to feed them and to change the cages and everything like that.
Mhm.
So I don't do any of that work. I was doing something that I had already had going,
Yeah.
but I finished.
Let's assume quarantine wasn't happening. What's your like normal kind of day to
Oh
day?
yeah, so when it's not quarantine, yeah.
Yeah.
So I'll come into the lab at like maybe between 9 and 10, something like that, and then sometimes I'll have something going from the night before. So like if I have an experiment that I set up the day before and I had let it go overnight, I would jump in and just start the next part. So that's like I put on the lab coat and gloves, and I'll add chemicals to things and move things around. And then sometimes I also do a lot of cell culture work. So So we have incubators and like a tissue culture room. So I'll go in, if I have something growing, I'll take it out of the incubator and I'll put it under a microscope and just see how the cells are growing. If they look healthy, if they are too crowded or not crowded enough, if they're growing in clusters. So just checking the status of the cells. And if something needs to be done, like maybe they need to be fed, I'll do that. And then like that's like I do that in a sterile hood. So like I'm sat with my arms in one of those giant wind benches. So yeah, so it's mostly I would say like on a typical day, maybe 80% lab work and 20% computer work. And then sometimes it kind of goes in waves, I guess, in cycles. So like sometimes I'll have a lot of experiments to do. So it'll be a lot of bench work where I'm actually doing the pipetting and all the wet lab work that you typically think of when you think of lab work. And then other times I've finished those experiments and I need to analyze the data. And so I'll just be at the computer. And so I have like I have a desk in the lab, and so I'll sit there. And so like a lot of some days I really don't even need to come in. Really, I could just do all computer work, and so like, that's what I'm doing right now, pretty much, because there's plenty to do. And so like I developed an app as well, because I designed this experiment, and the results from that experiment they're in like this in kind of like a non-typical format and just like the way that you have to think about it. So like you get results and then you have to think about what do those results mean, and that's where like statistics come in. And so in my situation, the typical analytical pipeline, like the typical statistics, are not really the best match for what the information is I want to get out of this experiment I've done. So I developed an app to just kind of help analyze that data. And so now I'm just like, I can make it better. So yeah, so it's like I have projects going in both the wet lab and the dry lab. So the dry lab is like the computer stuff, the bioinformatics, and then the wet lab is the actual bench work.
Cool.
So I do both of those.
So do you do like when you so you're this is I wasn't planning on asking you any questions about this, but I guess I will because this is fascinating. So you actually do you like come up with your own statistics then?
So like okay, so we have so basically what the app is doing is you have like in statistics you have a p-value,
Mm-hmm.
right? So like if a finding typically is under zero point zero 0.05 p-value, it's statistically significant. um, and so there's like some contention about whether that's good or not, but that's what is like typically accepted in the fields. And so if like you want to publish in any of these big name journals, which is kind of like publications in
Mm-hmm.
prestigious journals is kind of what scientists are going after for the most part. That's the end game, really. Um, it's not the end game, but it's like a milestone, basically.
Yeah.
Um, so so you need that p-value of 0.05 to get accepted to the publication. So like people have to do that. So, but with the experiment that I have, it's something called proximity dependent labeling. And so, as I was saying, we do protein studies and like looking at signal transduction. So the way that protein signal transduction occurs physically is the proteins come together in a complex, and then when they come together and like something else comes in, they change conformation, and that conformation change is what allows the signal to be transduced. So the signal itself is the actual, it's a cascade of proteins that A goes on to complex with protein B and C, but then A changes conformation and then is able now to interact with protein D, and then D goes on in
Is
you
that
know
just to
the
give
cascade signal.
just to give some of these some of the listeners like some context? Is that kind of like what you learn in like AP Bio with like electron transport chains and like photosynthesis? And is it like the same general idea where a change in one protein like adds something onto a second one and that?
Yeah, it's kind of like it's kind of a weird thing because it's underlying everything that you'll learn about molecular biology.
Mhm.
So like I don't remember in my schooling if anybody sat me down and straight up said like hey, the way that things change in the cell, like when a cell goes from being quiet to dividing, there's actually a process of physical interactions
Mhm.
happening in the cell, and those physical
Right.
interactions are between proteins. And so, like, I don't think it ever really struck me until I actually started, working on it in particular. Now I think it's fascinating. I mean, like, but
Mhm.
I remember not really knowing, like, or not knowing well. I mean, of course, it
Mhm.
makes sense, but you don't really think about it.
Right.
What's physically underlying, the electron transport chain. So, like the electron transport chain, that's a cascade of signal transduction. You
Mhm.
go from protein complex 1 to complex 2, and so it's signal transduction. So what I'm studying is signal transduction, but in the context of cancer. And so so in order to look at that, um, we're looking at the protein complexes themselves, because that's where the actual signal transduction occurs. And so we're using something called proximity dependent labeling, where we can take a protein of interest that we study in our lab, called uh, Abelson interactor 1. So we call it ABI 1 or ABI. And we have attached another protein to that protein. So we made a fusion. So it's like in the central dogma of biology, you have DNA makes
Mhm.
RNA makes protein. So we have changed the DNA, or we've actually synthesized DNA that expresses the protein that we study, ABI1, and then like a linker region, and then this biotin ligase called turbo ID. So a biotin ligase is an enzyme or protein that attaches biotin to things. So biotin is a small molecule, and it's special in that biotin is what is in eggshells. It's in eggs. So that's like it's the strongest non-covalent bond that exists in nature, between biotin and streptavidin. And so we attach this biotin ligase, this enzyme that attaches biotin to different proteins, to our protein of interest. And then we express that in cells. So now we have a fusion protein. So our protein of interest with biotin ligase attached to it. Anywhere the protein of interest goes, and whatever complex the protein of interest takes part in, we're going to have this biotin ligase floating around it. And so any protein that comes within a 10 nanometer radius, so that's like about the size of three small proteins, so you can imagine in a protein complex scenario. So any protein that comes within a 10 nanometer radius of our protein of interest will become biotinylated by the biotin ligase. And so we express that in the cell, and we break open the cells, and then we take all the proteins that were in the cell and we run it over beads that have streptavidin attached to them. So all the biotinylated proteins will attach to the streptavidin on the beads. So now we've enriched our sample for biotinylated proteins. And the biotinylated proteins in that sample in particular are the ones that came within a 10 nanometer radius of ABI1. And so we use that technology and then like our controls to identify, and then we so now we have all the biotinylated proteins enriched on the streptavidin column. We remove it from the streptavidin column, and then we submit it to this machine called mass spectrometer, and that is able to identify, based on the peptide sequence of the proteins, which protein it is. So you're able to, blast it through this machine, and it'll say, okay, you have this much ABI1 in your sample, and you have this much GAP-DH in your sample, and you have this much of whatever protein, keratin, in your sample. Um, and so we're able to use this technology, mass spec, in combination with our proximity labeling, to actually identify the interactome of ABI1. And so the interactome is all the things, all the proteins that ABI1 is likely to interact with or to be participating in a protein complex with. And so what I'm doing is, I mean, I'm using that tool, and then I'm doing wet lab experiments to validate our findings from it, but at the same time, I'm using a lot of dry lab tools, like a lot of bioinformatics analysis on the computer, to really deconvolute the data, because you can imagine it's so twisted and so much information. We have a list of 5,000 proteins that we've identified, and narrowing it down to which ones are the most probable interactors, which ones are the most exciting, and what's going to be the easiest to show, really, that we have identified it. And so this is all focused around the idea that if a protein is interacting with something, it's likely that it's affecting its function.
Mhm.
So, and we're this protein that we study, AB1, we've shown that it's downregulated in blood cancer, and in particular in the stem cells of blood cancer patients. And so we hope to use the interactome of AB1 because we think that it's acting as a negative regulator. And so the idea is that if we can understand what AB1 is interacting with in these stem cells, and especially stem cells from sick people, then we can find complexes that are critical to developing blood cancer, and we can drug those complexes, or we can drug targets that reside on those drug complexes, on those complexes. Yeah. So
Wow.
that's sorry. What
No,
was the question?
that was. I don't know what the question was, but that was phenomenal. I think that was great. I'm gonna. Wow, that was. We're studying genetic engineering as a class right now, so I think I'm gonna pull
Oh,
that.
sweet.
Yeah, I'm gonna pull that little snippet out and make all my kids watch it, even though they won't understand it, just to get like a.
All right, cool.
Yeah,
I
no,
mean,
yeah.
if you give me like a little tidbit about what you're teaching in genetic engineering, like I
Uh-huh.
can probably come up with some stories and like
Maybe. Yeah.
some examples.
Samples.
So
Maybe. I
yeah,
let me ask the rest of the questions I wanted to ask you
yeah, sure,
first.
sure.
Yeah, no. That was amazing. Thank you. It was
Oh,
just for my own
thanks.
personal edification. I learned a lot in the last 10 minutes. That was dope.
Sick.
Okay, so now like a much more mundane question. What kind of education do you need to get to where you are right now?
Yes. So I got a bachelor's degree, a bachelor's of science in biochemistry and molecular biology from UC Santa Cruz. And so
Banana
the
slugs?
banana slugs, that's right. So the kids in your class, they're uh, uh, middle school or high
High
school?
school, right? Like the
They're high
beginning
school.
of high
Okay.
school. Yeah.
Okay. Yeah. So I guess like for science, for me, it really started in high school actually. So we did like a genetics unit in my sophomore science class, and part of that was running gel electrophoresis. And so like the end result of that is that you get a band of DNA. And I thought it was the coolest thing that you could actually like physically see the band of DNA. Like that was super neat. And then we had this class in my high school called biomedical science, and they had like been donated some machines and stuff, like PCR machines, and so like, we could actually do a lot of molecular biology experiments in class. And so that like I was super into that, really kicked it off for me. So that's why I went to biochem and molecular biology at UC Santa Cruz. So I kind of knew the direction that I wanted to go into when I got to college, um, but I'm really solidified in like my junior year of high school, and like really from there. So during my university, in my third year, I did a study abroad, and in one semester of that study abroad, I just worked in a lab. So like instead of taking classes, I was just doing a bachelor thesis project. So that was really cool. And like I'm bringing that up because in my I guess like career so far, experience has been like the number one thing, like experience and really networking, like knowing people, like in terms of finding like getting a job and even like getting into grad school now. So that's why I'm bringing up these different experiences. So after college, like so I did the study abroad my junior year of college and got back in senior year. I jammed out all my classes because despite having the whole year study abroad and like taking classes for the first semester, and then the second semester counted as it should count as a class, but it was like a lab project, a thesis project. I didn't get any credit for the whole year
Wow.
from UC Santa Cruz. So I got back and like jammed out summer. I did like a full. I think I did 19 credits in the summer session, and like I was staying in Santa Cruz only 3 days a week, so I'd like drive down on a Tuesday, like go to Safeway and pick up things to make 4 sandwiches, and then just like crash on the couch at the Pearl House, um,
The Pearl
and then
House is
drive
where
back
a
on
bunch
Thursday.
of our
Yeah.
friends used to live, just for the record. But
Oh
continue.
yeah. Sorry. Yeah. Yeah. So basically, like my senior year was really focused. Because also, like I had to take all my upper division courses
Mhm.
during my senior year because they weren't offered my junior year, which is when people normally take them. I basically just jammed out everything in the senior year, just focusing on graduating on time, and I did. And then I spent about 3 months after graduation in 2014 looking for a job. So during that time, I was like making phone calls, sending emails, applying Craigslist, wherever, Monster. So it was a lot, and it was super discouraging because nobody like I sent probably at least 500 applications, like without exaggeration. I'd like to say more, and I got maybe 3 or 4 emails back. So like, not even like even that's even counting like automated replies. So like 4 emails back, and eventually I ended up getting an internship at the Buck Institute for Research on Aging, which is in Novato, California. So that's like just um, it's like maybe 40 minutes away from Richmond. I would
Okay.
say something like that. Um, and so I worked there for like a month and a half or so because I had actually gotten an interview from one of the contacts I had while I was job seeking before I got the internship. So during my internship when I was there, like I learned about like worm work and doing uh, some other like yeast work and stuff like that. And I was doing yeast work in Copenhagen as well when I did my study abroad. And I did a bachelor thesis project as well. So that was great. I didn't talk about that at all. About like what I actually did. Um, yeah, so I did an internship at Buck Institute, and then I got a job at UCSF. So that job, um, it was our family friend, their neighbor was a professor at UCSF, and our family friend asked the professor, like, "Hey, do you have any leads on a job? Because our friend over here is looking for a job in science." And he said, "Oh, you can contact this guy, and maybe he takes on students sometimes, so maybe you can do like a rotation role or like a research role in the lab." So like I reached out to this guy Frank Zoka at UCSF and said, like, "Hey, like I got your contact from so and so, and like, can I work for free in your lab? And like, please, like, just let me do something. I need some experience. I want to get my feet wet, and like, I'm desperate over here, like, looking for jobs." And he said, "No, like, we won't even let you work for free because by the time that we train you, it's going to take 6 months for you to actually do something useful, and then you're just going to have a job, and then you're going to leave." And so I said, "Oh
Yeah.
shoot, well, thank you very much. Like, that's disappointing, but I understand." And they said, "I do have a startup company that is looking for a research associate." And so he put me in contact with the 2 other guys from the startup, and I ended up interviewing with them at UCSF, and I got the job there. And so that was like really my foot in the door for research positions. Started the job at UCSF. A month later, I moved into San Francisco, and then I ended up living there for 3 years. I worked for this company at UCSF for a year and a half, and then they were a small company, so they were only 5 people, myself included. They were trying to make a drug for thalassemia, so they were developing this drug, and so like that's where I really got exposed to pharmacology for the first time. So like it was pretty cool, like being such a small company because like we'd be on like they let me sit on phone calls with the FDA and like doing like their updates for where we were with the development of the drug because they had been funded a bit by the FDA through this pilot program or this grant project. Um, yeah, and so I got laid off from there because they didn't make the funding goals that they needed. And the thing was with this startup UCSF, they have an incubator space. So that's like a lab space that multiple different companies work out of. And so one of the other companies that was there, they ended up like moving. They got funded, moved out about maybe 6 months before I got laid off. And so when I was looking for a job, I saw they had a post on Craigslist for someone that could have been me. and so I applied for that one because I had like a month in my apartment before I had to move back home,
Mhm.
and uh, so I just like was just applying for jobs and playing video games all day for that whole month, and like just being really really anxious about getting kicked out of my apartment. Um, yeah, so I ended up just sending off emails to those people I knew, and then texted them all and said, "Hey, like, look at my application. Got interviewed, got that job. Worked there for a year and a half, and then decided I wanted to go to grad school. And I, I think I said I want to go to grad school, and I looked up like when the GRE was because you need to
Mhm.
take the GRE. That's like the SATs for grad school. Um, and there was one at the end of the week, and like I had like a GRE prep book that someone at UC Santa Cruz had given me like at graduation. I like never cracked it, and so like I would just come home from work every night and just study for a week, and then I took it that Saturday, and
Wow.
I got the scores that I needed to go to programs that I wanted, and then like the next month or two was really just spent kind of like doing research on all these different programs, grad programs, and writing writing the letters and tailoring them to each one and looking up the professors that I might want to work with and yeah, because like I didn't have good grades at UC Santa Cruz. Like I think so. I had like a 3.3, I think, or 3.27. So like
Just for the
that's
records,
not.
guys, that was much better grades than I had.
Uh,
So don't
yeah, I mean, but for
don't
going to
disparage that.
grad school,
Yeah, no, that's not
yeah,
great.
I know,
You need
I
like
know.
a
Sorry.
4.0. Yeah, no, yeah.
Yeah, I know. It's kind of a dick thing. Sorry. Yeah,
No,
that's
no, no. It's all good. I'm just messing with
yeah.
you. You get
Yeah, so I didn't have like even now it still kind of bites me because like a big part of grad school is trying to get your own funding. So like
Mhm.
you want to get government organizations or private organizations to give you money to pay your salary and everything like that. Um, and the great thing from undergrad, it
No,
still
it's
comes
following you.
to bite me.
Wow.
Yeah, because even though like to me that was so long ago because I have all these different work experiences
Hmm.
that I mean they speak enough to get me to grad school, but you know, they're still lingering.
Those numbers
So
on those pieces of paper.
yeah, so
Oh,
it
that's
is it
rough.
is important, unfortunately.
Yeah.
But yeah,
You hear
yeah.
that,
So I
kids?
think
Don't spend your
that's
time running around the woods. Go to class.
that's. I mean, you could run around the woods. If that's what you're gonna do in your free time. Go do that instead, and don't sit around watching TV.
Yeah, fair enough. Touche. Um, awesome. I got like three more questions for you.
Yeah, sure,
Thanks for hanging in there. Uh,
not a problem.
what are some things about your job that you find, or like your day to day experience that you find challenging or difficult?
Oh, so sometimes you just get in a rut where things just don't work out.
Hmm.
Like you'll have contamination in the incubator, and so like all your experiments will become contaminated for 2 weeks before you figure out what's going on, and then you have to find the root of the problem. And you do a lot of science to figure out what's going wrong so that you can do science. So it
Hmm.
feels tedious, but it's something that you have to do in order to make reproducible results. Because that's what it's all about. Like we're here to try to improve human knowledge, and if we have something that is apparent but it's not robust, like so it looks like it's right but it actually isn't right, we just don't realize that it's not right. That's actually detrimental. It's worse to have that existing than it is to not have it at all. And so it's really important that we maintain a clean lab space and we make sure that all of our materials are good and the methods that we're using to do the things that we want to do are good. And the way that we check that is through running good controls. And so it's a lot of it feels really tedious, but for that's what is like the difference between you know I think doing this kind of science and doing backyard chemistry or mad science or whatever. It's you're really trying to make sure that not only can you do something, but can you do something in a way that other people can do it as well? Are you finding something that is like a neat little trick, or are you finding something that is an absolute truth? And that's what we're seeking. And so um, it can be tedious to kind of maintain the conditions that you need to produce good data, but but it's absolutely necessary. So I think that is my answer to your question: is all the things that you need to just keep organized and keep in order, or it can get kind of boring and feel overwhelming sometimes.
Thank you. And then last question: what do you like about
Oh,
your job?
just
Or
I mean,
what's pick the thing you like the most?
yeah. I like the aha moments. So like when you make a connection um, or you see a result that's what you're expecting, or even if you see something that you're not expecting, but you can make sense of it. Yeah. So like for an example, I made a cell line where I made this fusion protein. So the way that we check if the fusion protein is there or not is through gel electrophoresis. And so gel electrophoresis, it separates the proteins based on how large they are. And so and then you use an antibody with something that lights up attached to it, so that it only recognizes the protein that you're looking for on the gel. And so I made this fusion protein. So I have the wild type that is only expressing the regular protein, and then I have the fusion protein, the cells that are expressing the fusion protein, which is it's like double the size of just the wild type protein itself. And so like I ran it on the gel, and then I see like the perfect shift. So like one is down here, and then the other one is up here, and it's exactly what I expected. And so I think that's what I like the most. So it can be anything. It's maybe it's kind of like incremental uh, kind of reassurances, I guess. But sometimes they can be large. But either way, they're very very satisfying, and that's what I like most about doing what I do.
Awesome. Well, thank you so much for talking with us today, Max. That was super cool. That was
Absolutely,
fascinating.
a pleasure.
uh, maybe we'll have you on again sometime. Uh,
Absolutely, I would
yeah,
like it.
awesome. Listeners, if you have any questions, feel free to drop them into the comments and or contact me directly. I'm happy to forward them on to Max. Um,
If anybody wants my email too, Michael, you can send it to them.
cool. Sounds good. If you guys want Max's email, if you're somebody I trust with Max's email, I'd be happy to
Right on.
give that to you. Knuckleheads, that's you. I'm not giving you Max's
No
email.
funny business out
Yeah.
there.
But any of you nerds, if you want Max's email, I'm happy to hook it up. Alrighty, thank you so much. Let me just turn the screen capture off. Cool. Hey, real talk, Max. Thank you. That was super cool. Like just for my own. That was super cool just for me.
Oh yeah, I love talking about it. It's like
Yeah.
it's so nice to come across someone like you who's really interested